In this video, we will discuss about this SRC activity with PDAC progression and Dasatinib impact on the proliferation. And this Dasatinib impact on proliferation is also related to this SRC activity, as the researchers have checked with increase in the concentration of Dasatinib how this SRC activity is impacted by this increased Dasatinib, increase in the concentration of dasatinib. So let's start with the figure on the left hand side. So here what it did is they check this SRCC. What it did is they use the pancreatic cancer cells and infect the mice, and thereafter they check the pancreas at different stage. That is when it is at the first stage of the pancreas, that is pan L N1, and when it is at the pan N L3 stage, and when it acquires this PDAC stage, and they stain this tissues with this SRCC and the activated SRCC, that is phospho SRCC Y148. And what they are able to find out is that with increase in the progression of this PDAC, they see increased staining or increased signals for this SRCC, indicating that as the cancer cell, as this pancreatic cancer progresses, the SRC activity increases. And when they check the results for the activated SRCC, they are able to see that in the normal duct they do not see, or very minimum amount of this activated SRCC signals has been found. But with the pancreatic cancer progressions, they are able to see increase in the signals for this activated SRCC, indicating that the PDAC progressions has a direct impact with this SRCC activity. Thereafter, they wanted to check whether dasatinib has any impact on this SRCC activity, and the final outcome, that is proliferations, will also get impacted with this dasatinib or not. So in the panel A, they first shows the three different cell lines that they have developed by impacting this or by infecting the cells with this PDAC. So this is the mice numbers or that they have used, and these are the cell lines which they stain with PDX1 to shows that these are the pancreatic cell lines. And they also check this recombinant P53, that also further validate that they have these cell lines which is derived from the mice models. And then they check the impact of the dasatinib or dose dependent impact of the dasatinib on the activated SRCC. And we can see that with increase in the concentration of dasatinib, the activated SRCC signal goes down, and it goes down at a 200 nanomolar, it goes down to almost negligible. And these things they have observed for all the three different cell lines, that is for 82739, 83320, and also 86119. The trend follows the same things, that is with increase in the dasatinib concentrations, the activated SRCC goes down. And when they check its impact on the proliferations, what they are able to see is that there is not much impact of proliferations up to this 200 nanomolar concentrations that can be seen here. But when they increase the concentrations of the dasatinib, they are able to see some decrease in the proliferations to around 40%. And this decrease, they have hypothesis or assume that this decrease could be because of the impact of the dasatinib on the other factors apart from this SRCC. So till 200 nanomolar, they are assuming that this SRCC, this dasatinib has an impact only on SRCC, and but those SRCC impact does not cause too much decrease in the proliferations. And when it goes up to this 1 micromolar, the decrease in the proliferations could be because of the other tyronase activity that plays a role towards this proliferations. So this is what their assumptions for this 40% decrease that they see in the 1 micromolar concentration of dasatinib. So this is what they have presented in both two figures, that is SRCC activity with PDAC progressions and dasatinib impact on the proliferations and this SRCC activity. So I hope you understood these two results in brief, or you get some informations about these two results. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.